Good evening, everyone. Welcome to week 7 of the live interaction sessions for the Neptune Immunology course. This is also a rescheduled class, which unfortunately I had to do because of some unexpected works, commitments that came up. Since no one has joined the class yet, I will go ahead with the discussion so that it is available to you to access online at any point of time if you want to. And also the lecture will be uploaded on YouTube, and you will also get access to it via the Google Drive link that you would have for the course. So before we get started, let us just recap what we discussed in the last class so that we can directly go to the assignment for that purpose. So if you remember, for week 6, we had looked majorly at the techniques which are necessary for which involve the use of immunological concepts, basically meaning that which involve the use of antibodies. And the reason that they are utilized is because of their specificity, because of their ability to precipitate out of solution when they interact with each other, and their ability to recognize specific motifs within the cell. Uh, Not motifs, the actual word would be epitopes, and they are able to recognize those epitopes and detect them inside a cell. So we discussed multiple types of techniques, like starting off with radioimmunoprecipitation, the different subtypes of it. There was some extensive discussion about Western blotting as well. What is the technique? What are the requirements? I had some additional videos added for it as well. You can access that. Apart from that, there was some discussion on the use of antibodies for visualization. When I say visualization, it is with respect to the use in immunofluorescence techniques, which is often also known as ICC or immunocytochemistry, and also the point of distinction with immunohistochemistry, which is another subtype of the same. So with all this, we had basically looked at the techniques that involve the use of antibodies. Otherwise, they're also known as immunotechniques. So with this in mind, we will first look at the assignment questions so that that can be quickly sorted. And then we'll go ahead with the discussions regarding the same. So if you remember, for week 6, the assignment you had submitted it a while back, but because I did not discuss the questions, I will go through them quickly once. So the first question was about the radial immunodiffusion assay, whereby we were asked that the area of the precipitin ring is proportional to A, the concentration of the antigen, B, concentration of an antibody, C, concentration of both, or D, none of the above. So if you remember, we had seen how a graph can be plotted to see what is the how you can derive what is the concentration of the unknown antigen. So you basically plot a standard curve. You have some known concentrations of antigens, and you basically make small wells within an agar plate into which that antigen at different dilutions is added. And the entire agar is made up of antibodies. So wherever there is interaction between the antigen and antibody, precipitin rings will be formed, and that will the area or the spread of that precipitin ring will be an indication of how concentrated or how diluted that antigen will be. So based on that, we were able to plot a graph, a standard curve, where for each concentration, the area of the ring could be noted, and whatever was the area of the unknown precipitin ring, or the radius of the unknown precipitin ring, helped us in determination of the actual concentration for the same. So with this, the correct answer therefore becomes option A, whereby we look at the concentration of an antigen for this purpose. Secondly, we were asked that an exogenous protein was overexpressed by a student in some cell lines. Which technique would be best to determine the overexpression of this protein in the cell lysate? So it's a very obvious answer. I think it was something I discussed in the discussion session as well. So basically, whenever you overexpress a protein, it means that you are adding some sort of construct, which is construct basically means a plasmid under most circumstances, which is which carries that gene under a very strong promoter or under the control of a very strong promoter that allows it to be transcribed and translated at very high levels within the cell, leading to an overall elevated expression of that particular gene or that particular protein inside the cell which we are studying. So, therefore, whenever you want to analyze that or you want to check that there has been actual overexpression of that protein, the technique you go for is Western blotting. So, I think I had mentioned it that Southern blotting was because of the discovery made by Dr. Southern, who who named the technique after his own name. So, thereby, the type of molecules that are studied are the DNA molecules. So basically, it is dependent upon the hybridization of DNA. When I say hybridization, it simply means the binding of DNA on a solid surface, which can then be probed and studied. The second type is northern blot. So northern was because it is the complete opposite of southern blotting. Northern blot was indicative of RNA molecular characterization or molecular understanding of RNA population inside a cell. That was for northern blot. And thirdly, western blotting, because it was completely different from these two, was dependent on the movement or migration of proteins that have not migration of protein, or let's say the hybridization of proteins onto a charged membrane, which have separated based on their mass by charge ratio when they are run through an SDS page. So taking all of this, the correct answer becomes Western blotting, and that is the most common technique that is used even till date. You can use other methods, most commonly when you want to check overexpression of a protein which is significantly abundant in the cell. Then immunofluorescence based methods are quite extensively used. And it has been seen that basically what you will see is in a control cell or a untreated or empty vector cell, in a cell that carries an empty vector or the control condition for that overexpression, there you will see very minimal or a baseline level of expression, whereas in the overexpression construct carrying cell, you will see a much higher expression of the same. So that can be visualized using, fluorescence microscopy, whereby you'll be able to see a clear difference between the two. However, a better understanding of this is definitely understood by techniques like Western blotting. When you just want to check RNA in today's time, we do not go for Southern blotting. Instead, the more so to say cost-effective method involves the use of qPCR or quantitative real-time PCR. Whereas if you are doing a high throughput, if your objective is to go through a high throughput assay, then you opt for transcriptome sequencing, all of the RNA inside the cell to see what is the level of expression. To see what RNA is being expressed or what genes are being expressed at that time under that condition. And B, to characterize what is the expression level of that RNA in terms of, there's a different unit for it, which is called as transcripts per million, which is after normalization. You will get to compare what is the relative expression of a particular RNA with respect to some housekeeping RNA. So that is for beyond the scope of this course as of now. But basically, what we are trying to focus on is the use of antibodies in Western blotting. Third, in the immunostaining technique, which is a part of immunofluorescence or immunocytochemistry as we know it, paraformaldehyde is used as a fixing agent, B, permeabilization agent, C, blocking agent, or D, detection agent. So paraformaldehyde is actually utilized as a fixing agent. So what it basically does is it helps in preserving the cell in whatever condition it is. It helps in, to some extent, helps in formation of pores on the cell membrane, which will allow the entry of antibodies into the cell. Because if you think about it, normally a cell membrane is supposed to act as a barrier. So it would not allow or would significantly retard the entry of any foreign molecule into the cell, right? So to bypass that, what is required is the formation of some pores, which will allow its entry to be facilitated. So that is done by the fixing agent and is supported by the permeabilization agent. So permeabilization agent makes very large pore, or it enlarges the pores that have already been formed. That allows easier uptake. Blocking agent is for non-specific to block or to remove the non-specific binding, or reduce at least the non-specific binding of the antibody to any random proteins within the cell. And fourth is the detection agent, which you use for actually identifying where your antibody has bound. So the correct answer in this case is a fixing agent, which is option A. Then we had discussed extensively what is the purpose of blocking in ELISA. So if you remember the entire topic of ELISA, we had seen all the different subtypes, we had seen how they work, we had seen the substeps, the subtle differences between the different types of ELISA, how it is commercially very useful, where is it, like if you look at any path lab report, you will find some test or the other which does involve the, which does depend upon the concept of ELISA, which requires antigen binding and then antibody binds to that and so on and so forth. So, yeah, so the question is asking about the reason why you need to perform blocking in ELISA. So, A, option A is to promote binding of antigens, B, to enhance the sensitivity of the assay, C, to inhibit the activity of enzymes, or D, to prevent the non-specific binding of antibodies. So that is option D, that is prevention of the non-specific antibody binding. So it blocks any of those sites, any or all of those sites. That might be too strong a statement. Let's call it most of the sites where any non-specific binding can occur. Basically, the idea is that beyond that also we can expect that there might be some non-specific binding, because at the end of the day, it's an antibody and it will bind to any region which carries the sequence of the epitope or the structure of the epitope which it is specific for. So with that in mind, with taking into consideration the specificity, or that actually dictates how specific that antibody's action or activity will be, we can see that there will be a difference, and therefore to eliminate the discrepancies that you might see for antibodies efficiency when used in Western blotting, the objective is to perform blocking of those non-specific binding sites and prevent this from actually happening. Then we look at Then we look at question 5, whereby we were asked about certain statements regarding ELISA. A, it is a highly sensitive technique for detecting and quantifying soluble substances such as proteins, peptides, antibodies, and hormones. B, it uses antibodies that are labeled with enzymes that produce a measurable product when the substrate is added. This technique does not require separation of free and bound antigen. The color change in ELISA indicates the presence of a target analyte. It is possible to use monoclonal antibodies in ELISA. The intensity of the color that is produced is directly proportional to the color of the target analyte. These are some statements. What we've been asked is that which one of the following options contains a combination of only and all correct statements. So if you look at it, let's go over them one by one. The first statement was about the fact that it's a sensitive technique. What does it do? It detects and it quantifies soluble substances like proteins, peptides, etc. That is indeed the correct technique. It's not just about detection, because it is attached to a signal which can be monitored. Say, for example, when we talk about immunofluorescence as a technique also, or immunocytochemistry as we accurately often describe it, in that case, what you get as an output is a readout of the signal, which is present on the antibody that is being used for study. Say, for example, if you are detecting, um, let's say, actin, right? So what you'll do is you will probe that cell using an anti-actin antibody, and what those antibodies will do is they'll go and bind to that location, and then you detect where or which are the locations where that anti-actin antibody has bound. That detection can either be via the use of a direct tag. For example, if you have a tagged anti-actin antibody, which is not always the case, it's sometimes there, but it's an expensive venture and its utility gets significantly bottlenecked or narrowed down, basically. Alternatively, what is done is after the primary antibody is bound, the secondary antibody binds on top of it, and that is then attached to some signal, which is what is given as the readout. So therefore, it is extremely efficient in the quantification process as well. So statement A is correct. Second, we've been asked that it uses antibodies labeled with enzymes that produce a measurable product when the substrate is added. That is what I just explained, and therefore option B in this case would be the correct option as well. See, it does not require separation of free and bound antigen. That is not correct. Why? I'll explain it a little further. Say, for example, when you're performing ELISA, in case you have antibodies that do not, like if you have a population of antibodies, if it is not just monoclonal antibody which is being used, and what you will observe is that you will have a solution of let's say actin antibodies, but some subpopulations of them can actually be grouped together or can be further classified to find the class that are binding to one particular epitope on the on the actin protein or to another epitope or so on and so forth. So if that is the case, then you should keep in mind the fact that if that antigen is now bound to something, say for example, actin is bound to phalloidin. In that case, what will happen is that if this is suppose actin and phalloidin binds on top of it, then this entire area where these two interact with each other is blocked and no antibody can now access it any further, which is a huge problem. And therefore, it was something that is usually avoided, and you go for A and B statements for now. In option D, we have been asked that the color change in ELISA indicates the presence of a target analyte. When there is a color change, which is something we do observe, you get the presence of target analyte. That is actually true. That is what you get as a result of quantification. So when that analyte is present, antibodies are binding, there is a shift, and what you get as a readout is what you monitor. Then in F, the intensity of color produced is directly proportional to the concentration of the target analyte. How intense is the color which you are getting in this case? That will be proportional to how concentrated the target analyte actually is. So based on that, our correct statements eventually come down to be A, B, D, and F. So these are our correct statements, and therefore the correct option. Okay, I somehow got muted. Apologize. So we're saying the correct option in this case could be option D, which marks these statements A, B, D, and F as the correct answers or correct facts about ELISA. Then we have been asked for the correct sequence of Western blotting. So immunoblotting or Western blotting is something we have extensively discussed now in the last class. In case there's any confusion, you can go back to the PPT. I have mentioned a lot of details there, and you'll be able to brush it up really quickly also, I think. So yeah. So we've been asked about the correct sequence. So first is about performing the page or the poly SDS page or a non-denaturing page, which is polyacrylamide gel electrophoresis. Literally meaning that you are using a polyacrylamide gel and pushing current through it to allow the separation of proteins. That's what the technique does. Once proteins are separated based on their mass or their molecular weight, as we now know it, then what it does is, what you need to do is you need to transfer them onto a PVDF or a nitrocellulose membrane. Therefore, that becomes a step. Yeah. Okay, I think there is some. Okay, we'll just read out the statements first. That is the transfer of the proteins. The third statement is about the proteins being denatured by SDS and by boiling. Now, as you can obviously understand, you cannot denature proteins or boil them after they have been run through a polyacrylamide gel, right? Or they have been subjected to page, basically. So that means this becomes the first step, whereby first proteins are denatured, they are boiled. In a large number of cases, which is something we at least do and is actively practiced, is the addition of beta-mercaptoethanol, which is a reducing agent that allows a reduction of the disulfide bond present between cysteine residues and helps in the production of cysteine, which means there are two cysteine residues that are linked by a single bond, and they might be present between amino acids which are hundreds of bases apart. So if that is the case, then that helps in some specific structural modifications, and therefore it is speculated that if in case that structure is retained within the gel, then what it will do is it will retard the movement of proteins through the gel. So to eliminate that possibility, what you do is you add beta-mercaptoethanol under most cases. So whenever a reducing condition is not a problem. Say, for example, if you want to get some functional enzyme as the output, then in that case, adding beta-mercaptoethanol can actually be dangerous. It can, affect the overall structure and therefore should be avoided. So in step 4, we were asked that you incubate the PVDF membrane with primary antibodies and secondary antibodies subsequently. So that is certainly a correct option, and that happens fairly towards the end. So basically, what you will first see among all of these is that the protein needs to be denatured by SDS, and then it needs to be boiled. Second statement is that it needs to undergo the electrophoretic steps or the gel separation of proteins based on their molecular weights through a polyacrylamide gel. Then we have the transfer of those proteins. Once they have separated, you want them to be transferred from the gel that is being taken into consideration onto the membrane, which you can now use as a solid support. And then finally, you incubate it with the primary antibody of interest and the secondary antibody of interest subsequently, and then utilize it further. So then our correct option in this case becomes option C, which is 3, 1, 2, and 4. That is the sequence of events. Then we were asked that in the immunostaining technique, the proteins are stained by A, the antigen, Kumasi brilliant blue, silver stain, zinc stain, antibody conjugated to a fluorophore, or radioactive probes. Now, while the correct option, um, no, while the first option might seem very tempting to you, and you might think, yeah, proteins are getting detected because of their antigen only. But that's not actually the correct answer in this case, because here we've been asked what they are stained with. So that staining part, their detection can be because of the presence of the antigen. You are using a certain antibody, which is allowing this to function in its own manner. However, when we have specifically been asked how they can be stained, then instead of the antigen, massive brilliant blue, silver stain, zinc stain, and radioactive probes, the correct answer would be antibodies that are conjugated to fluorophore. And this is mostly, once again, done majorly for the secondary antibodies, so that you would be able to cover a very large number of proteins, and you'll be able to do so very efficiently. So in this case, the correct option is that antibody should be conjugated to some sort of a detection signal. That can be a fluorophore, or that can be some luminescent signal, etc. Most commonly uses fluorophore. Alternatively, what is also often done is the use of ECL reagent, which are electrochemiluminescence reagents. And basically what happens there is, I think we'll skip it for now. Let's come back. But I can just briefly explain it to you. So you can look up the enzymes that are involved there. So it basically consists of a reagent. You have two types of reagents. You have an enzyme and you have a substrate. You allow them to interact with each other, and as a result, you will see a difference. So in this case, the correct option was option E, whereby antibody was conjugated to, or has to be conjugated to, a fluorophore. Then in question 8, we have been asked that in flow cytometry, what does light emit as a forward side scatter measure? So if you, I think you might be remembering. So if you remember, yeah, I don't think I went into much detail of it, but it was there in the lectures, whereby forward scatter and side scatter were explained. They're very, very crucial terms in terms of, IP, or flow cytometry, etc. Not IP, just flow cytometry. So there you can see that we have been asked, what does FSC measure? Options given in this case are cell size, cell complexity, cell surface biomarker, or all of the above. So in this case, the size of the, or the dimensions of the entity of this cell or the droplet that is being studied, that did not get factored in. So yeah, in this case, the correct option is cell size only. Size complexity doesn't matter. Its fluorescence doesn't matter, etc. In fact, cells are sorted by a dilution plating until there are only single cells in each microtiter plate well. The differential weight, electrostatic force or magnetic force. So cells get sorted because of the electrostatic force, which allows it to be segregated based on the surface markers that it carries. And finally, in question 10, we were asked that in the immunodiffusion test, if two lines cross each other, then what does it indicate? A, that they are identical, B, they are not identical, or they are partially identical, or none of the above. So we have been asked what happens if if two lines they cross each other from both sides of the antigen, then what exactly is to be done? So basically you will consider that they are not identical to each other, and as a result they are not retarding each other, and therefore option D is the correct option in this case. So with this we come to an end for the discussion of the week 6 assignment for this course. Now, before we start, I'll start in just in 2 minutes. Please excuse me, I'm getting a call. Sorry about the disruption. Sorry, I was getting a call. Okay, so now we'll quickly go over a revision of this week's. And I just need to make this. Yeah. So I have not completely like repeated what has been taught by professor in this in the YouTube lecture videos. I hope the slideshow is visible. Okay, so what I've basically done is that I have included some of the experiments that were not exactly discussed by Professor Shrudeep in the lectures, and try to take again everything that I have mentioned that I will be mentioning today has been taken from Kubi Immunology, and you can refer to it if you want to read the experiments in more detail. If you have any queries, you can bring them up. I would really encourage and would really like to have a live session again with the students because we had a good rapport going. But yes, it got disrupted because of my schedule. So again, apologies for that. But I'm hopeful that it can again go back to the regular schedule that was maintained on Sundays in our regular classes from 3 to 5. So coming back to the presentation. So basically today, the idea is to majorly center around T cell development, T cell mediated immunity, T cell signaling, how that is controlled, etc. So starting off, this figure have attended the more initial lectures. Sorry. More not lectures but the discussion sessions. So again, this is from Kubi, like I mentioned, and what we had seen here was the sequence of events that occurred that led to the development of T cells in the mouse. So basically I'll just revise it again because it's relevant to our topic for this week. So what we had started off with was a progenitor or a very precursor cell, which is considered to be the starting point for T cell development. So that is, of course, the hematopoietic stem cell, which is present within the bone marrow. Now, from the HSC or the hematopoietic stem cell, we can see progression through multiple stages, starting off with the common lymphoid precursor that is from the hematopoietic stem cell, which can potentially give rise to any cell type. They could have given rise to any cell type eventually. From there, it undergoes some fate assignment, basically, that it is now destined to become a cell of the lymphoid origin, and therefore it is working as a common lymphoid precursor. So there we see the maintenance of surface markers like c-KIT, CD25, CD44, etc. Then we see migration through the blood, followed by presence within the thymus. What are the events that are occurring within the thymus? Let's go over them. So first of all, we see that there is a T cell precursor. Upon migration via the blood into the thymus, we are seeing the presence of a T cell precursor, from where we see a pro T cell, that is a double negative variant of the T cell. You are now I assume, quite familiar with the concept of double negative, single positive T cells, double positive T cells, etc. So while this transition is occurring from the T cell precursor to the pro T cell, which is double negative, how is it actually distinguished? One might ask that if there is no new antigen, when we say double negative, it means it is not CD4 positive or CD8 positive, which means it lacks both of these surface markers or surface antigens. Then how would you actually distinguish it from the T cell precursor? First is that you see a gradual decrease or dissociation or the disconnect from surface markers like c-KIT and CD44. In concordance, what you see, an event that occurs that actually distinguishes these two substates, is that there is locus rearrangement, or there is rearrangement at the TCR locus, that is the T cell receptor locus. So what happens there? You see that there is expression of the RAG gene, which is a recombination associated gene, which is a recombinase, basically, that is able to cut DNA at some distant locations and allow there to be a joining of some other combinations of DNA, eventually leading to the formation of that recombined locus. So there you see that initially the TCR locus rearrangement is D beta and J beta rearrangement is occurring, and then when that rearrangement has occurred and there is a simultaneous, gradual decrease in c-KIT and CD44, basically what you will consider that is to be a pro-T cell. After this, after D beta J beta joining has occurred, there is another round of recombination attaches it with a definite V beta region. Now, if you remember or if you've gone through the lecture notes or basically the YouTube video lectures of Professor Shrudeep, you would remember that the way in which this actually occurs is that there is a sort of permutations and combinations are tried, whereby you try joining a certain, say V5 is being joined with a T and J, that is downstream, but that is not leading to a functional rearrangement, and then that will not be entertained, and such a cell will eventually be lysed and eradicated from the system. If that doesn't occur, there is the formation of a very, very clinically relevant technique, then that is the emergence of the pre-T cell, which is also double negative, but now carries a V beta fusion with D and J beta. So there has been attachment of the variable region, and that leads. There has been attachment of the beta region to the D beta J beta region, and that leads to the formation of the double negative form or the double negative variant of this stem cell. Following this, there is attachment with the alpha chain. So if you remember, once again, reiterating what we have already discussed, that TCRs or T cells can be of two types. Either they would carry the alpha beta chains, or they'd carry the gamma delta chains. And as a result, based on most examples that we'll be studying, will have the alpha beta rearrangement. And because this has been studied in mouse, the most prevalent one is what has been demonstrated here schematically as well. So when that happens, you're able to see a transition, or we can say a change in nomenclature rather, from the pre-T cell to the pro-T cell. It is still, you can see that most of the things are similar, that there has not been much shift in, expression of other genes. But what you see here, A, is that there is a combination between the alpha gene and the beta gene, which is a major change that is occurring. Second, you can see that there is a shift in expression of certain genes. What are those? First of all, if you pay attention here, you will see that there is a very small, it can, okay. So, first of all, what you see here is a very small molecule at the cell surface, which is actually the pre-alpha chain, otherwise denoted as pre-T alpha. Following that, you can see that there is the appearance of two colored surface antigens. And if you remember these from your very, I think in week one, most probably we had this discussion where we distinguished the different subtypes of T cells, and we had looked at both CD4 and CD8 positive T cells. Then you would remember that these are different entities. The pink one is representative of the CD8, and a blue is representative of CD4. What are these? Why are we calling them CD? Basically, they are surface protein antigens under most circumstances. In some cases, they can be lipid molecules, glycoproteins, etc. What effectively they do is that they are allowing clustering of these cells because of the presence or absence of this particular antigen. So basically, you are getting a category based on which you can separate these cell types. And now they are helping you in generating clusters that can be differentiated because of the presence of that particular cell type or that particular antigen. So they are allowing you to generate clusters that can be differentiated, and hence they have been named in that manner, that they are CD molecules which are present on the cell surface. So CD4 positive and CD8 positive phase come much later. So it goes from the double positive to this single positive state, and then it can migrate via blood into the peripheral tissues, and then you get these single fated cells. So that is the overall process which takes place. Second, what you see here is a graph which sort of manages, or it gives you an idea of the time course or the schematic timeline which is followed. So what you see here is a plot that shows days of gestation plotted against CD3 plus or CD3 positive cells. So what you see is the time course of appearance of gamma delta thymocytes and alpha beta thymocytes during mouse fetal development. So the percentage of CD3 plus cells in the thymus that are double negative, that means they are CD4 minus and CD8 minus, and bear the gamma delta T cell receptor shown in black, or a double positive and bear the alpha beta T cell receptor that has been shown in blue. So in mouse fetal development, you see that in the initial stages, what you see is the percentage of these cells is quite high. Initially, approximately, I would say somewhere around 85%, 85 to 90%. And then that abruptly goes down somewhere around the 18th day of gestation. That is around 2.5 weeks. However, when you look at the alpha beta thymocytes, initially there's no expression of these thymocytes and they're not necessary. However, somewhere around the 16th day, you can start seeing a gradual rise and then an abrupt rise from the 17th day in the level of expression. And then that keeps on increasing and is maintained in the adult. So based on the fate of each of these T cells, there can be a difference in the prevalence of their expression levels within the individual's body as well. So that was just a time course. Now coming to the details of the process. So this is what you see schematically, a representation of the pre-TCR or the pre-T cell receptor. And you can see how it has actually been formed. You can see all the different types. So this is the structure and the activity also of the pre-T cell receptor, also known as the pre-TCR. Binding of ligands is yet to be identified to the pre-TCR, is helping you in generating intracellular signals that can induce a variety of processes. So what you see here is that there is a pre-T alpha chain and there is a TCR beta chain. The pre-T alpha and the beta chain, or TCR beta chain, are all together or taken together are forming the pre-TCR. What happens there? When they are able to bind to each other, they are generating signals which allows a few downstream processes that are being represented here. The first being that the cell now becomes permissive for TCR alpha chain locus rearrangement. So once you can actually validate the fact that alpha pre-alpha and the complete TCR beta genes are able to interact with each other and they're able to generate a potentially functional protein, then the cell will basically give the green signal to go ahead and proceed with alpha chain locus rearrangement as well. Secondly, you would be able to, if these interact and they are able to form a functional receptor, then they would stimulate the expression of CD4, CD8 co-receptors. Then we have the next downstream expression is that it would stimulate proliferation. And fourth, it would stop additional TCR beta chain locus rearrangement by a process known as allelic exclusion. Now, I have not included it in today's class. Maybe we can do it sometime towards the end of the course, or maybe probably when we discuss cytokines, maybe in that week. It's a very interesting concept, but I'll be honest, it's slightly advanced. It's a little difficult to grasp on the first try. So, maybe we'll wait it out a little while. So let's just go through the figure legend for our reference. So most double negative thymocytes, they progress down the developmental pathway. They stop proliferating and instead they shift their focus towards rearranging the TCR chain genes. What does that do? That helps in expression of the TCR beta chain genes, that then they can express that. Those cells that fail to productively rearrange and express these new chains will die. Newly synthesized chains will then, alpha chains will then be able to combine with the newly rearranged beta chains will be able to combine with a 33 kilodalton glycoprotein known as the pre-Ta alpha chain and associate with the CD3 group to form a novel complex called the pre-TCR. Some researchers have suggested that the pre-TCR actually recognizes some intrathymic ligand and then is able to transmit a signal through the CD3 complex that activates signal transduction pathways that may have several downstream effects. So basically what they're saying is when the pre-TCR, it recognizes some intrathymic or some antigens present within the thymus, and then their signal transmission is carried out by the CD3 molecule, or rather the CD3 complex that activates the signaling transduction pathways. It indicates that a cell has made a productive TCR rearrangement and signals its or allows its further proliferation and maturation. Second, it suppresses further rearrangement of the TCR alpha chain genes, resulting in allelic exclusion. Third, it renders the cell permissive for the rearrangement of the TCR alpha chain. Fourth, it induces developmental progression to the CD48 double positive state. So these are the different events that occur in the time course of T cell development after its productive rearrangement has been found. Now, this slide might look a little boring to you. I could not find very easy to interpret diagrams in this case, which is why I've not included it. But I'll try to, I just wanted to keep it there so that you can have an overview of the entire process from a bird's eye view. So I'll just get through that. It won't take long. We'll just read through it quickly. Excuse me. Okay. So what is happening here? We talked about the TCRs going through the double positive state where both CD4 and CD8 co-receptors are being expressed. What happens after that? Following this, the thymocytes begin to proliferate. However, during this proliferative phase, TCR alpha chain gene rearrangement does not occur. It is the beta chain which undergoes rearrangement, and we saw how it goes first from J beta, DJ beta, and then it influences alpha chain, and then it proceeds further. So both RAG1 and RAG2 genes, they are transcriptionally active, but the RAG2 protein gets rapidly degraded in the proliferating cell. Therefore, the rearrangement of alpha chain genes will not begin until the double positive thymocytes stop proliferating, and once they're able to proliferate, RAG2 protein levels will be restored, or let's just call it an increase. The proliferative phase prior to the rearrangement of the alpha chain increases the diversity of T cell repertoire by generating a clone of cells which carry a single TCR gene rearrangement. Each of the cells further can give rise or can rearrange a different gene, thereby generating a more varied or more diverse population than in the original cell had first undergone rearrangement at both chain loci before it proliferated. Okay. Coming to the next part, it is a very, very expensive process. When we say expensive, not monetarily, it's always in terms of resource for the host. So an estimated 98% of all thymocytes do not mature. What happens to them? What is their fate? They die by apoptosis within the thymus, either because they fail to make a productive TCR gene rearrangement or because they fail to survive thymic selection. Double positive thymocytes that express the TCR-CD3 complex and survive thymic selection develop into some immature single positive CD4 thymocytes or single positive CD8 thymocytes. These cells undergo added negative selection, and then they migrate from the cortex towards the medulla, where they pass from the thymus into the circulatory system. So that is the job. Now, these thymocytes can therefore, in summation, we can say that as part of the selection process, these thymocytes undergo two selection processes. First, there is a positive selection for thymocytes bearing receptors that are capable of binding to self-MHC molecules that allows for MHC restriction. This is the key word. The process whereby these T cells that undergo or that bear receptors that can bind that you are only allowed to retain those cells that actually have receptors capable of binding to our own molecule, MHC molecules that results in MHC restriction. Cells that fail positive selection are eliminated within the thymus by apoptosis. Negative selection, on the other hand, it eliminates and thymocytes bearing high affinity receptors for self-MHC molecules alone or self-antigen molecules presented by self-MHC, which results in self-tolerance. What does this mean? Let us use this. We will try to understand both these concepts. The first one is self-MHC restriction, and then there is self-tolerance. Okay, so the first term, you already are familiar with it from the lectures, I presume, but I'll just try to brush up your memory regarding this. When we say self-MHC restriction, it means that our cells, our T cells or our immune system needs to have some component which is allowing it to be, which is allowing it the ability to be able to distinguish between self and non-self. The way it is done in this case is that it looks at the fact that any cell, any T cell, which is able to identify my own body proteins and therefore understand that this is not something which is foreign and therefore not something that needs to be uh, removed from the system. It will not elicit an unnecessary response in this case. Cells that fail positive selection get eliminated within the thymus by apoptosis. Negative selection, on the other hand, it eliminates thymocytes that bear high affinity receptors for self-MHC molecules alone or self-antigens present by self-MHCs, which results in self-tolerant behavior. So basically, you look at two methods via which T cell selection occurs. We have, of course, got the group where there is under positive selection, what we see is that there is a restriction for self-MHC. Under negative selection, we make sure that any T cell which binds unnecessarily very strongly with our own self-MHCs, or in other words, with our own cells, that should definitely be removed. And that means that in that case, we will have a condition where it where it is tolerant but only towards self and is able to maintain that tolerance at a different level. So that's how that entire thing works. Just give me a minute, please. Okay. So what we see here is the experimental strategy that was used to understand the role of thymus in context of the immune system in mice. Now, it might seem very obvious to you when we talk about T and we emphasize that this is where they should have originated and this is how they should have worked, but that was not enough. As has been discussed by Professor Shudeep as well, basically what happens is what led to the initial discovery was that when this particular organ, the thymus, was removed from the mice that were under study, they saw that there was it led to a compromised immune system of such individuals. When I say individuals, I mean mice in this case. Therefore, it was understood that thymectomy or thymectomized mice basically can be understood to have a very compromised immune system. When we say compromised immune system, it is more of a generic term, and rather, if you go to any of the I would not take any names, but if you go to any of the sites where you can find, for example, some commercial companies that are able to maintain these mice with these definite genetic backgrounds, you'll be able to see how each of these immunocompromised mice are different from each other. So, for example, okay, let me just take some names. There are two major companies or major laboratories from where all of these mice are usually procured. One is called as the Jackson Lab. The second is Charles Rivers. So they are large multinational, not multinational, but multi-millionaire led companies whereby they maintain these strains of immunocompromised mice. If you go to their websites, you will be able to see that there is a huge range under the immunocompromised mice as well. For example, the one which is being studied here are considered as nude mice. Now, as was introduced already by Professor Shudeep in the lecture itself, that when mice are subject to a condition where their thymus is being externally eliminated from the system, it is leading to the condition where these mice are just immunocompromised. They do not have a functional immune system. When this was further dissected, what could be generated was a range of mice which lacked certain specific components of the immune system because of some genetic backgrounds that they carry. Say, for example, the most common mice which is used in cancer research is not-SCID family of mice. So not-SCID stands for non-obese diabetic, which is a variant of those mice, and SCID stands for severe combined immunodeficiency, like Sir had mentioned. These mice have a fur coat. They have a white fur coat instead. They are immunocompromised, but the extent to which they're immunocompromised can be quite different. They carry certain background mutations which will differ based on which mice you're procuring. Say, for example, the mice you get from Jackson or you get from Charles Rivers or you get from Indian companies such as Hylasco, for example, all of them will have different genetic backgrounds. Majorly, one common mutation is within the PRKDC gene, within IL2R gene, etc. So what that leads to is the fact that while not-SCID mice are immunocompromised, they still have a condition where their NK cells can work. When you look at super not-SCID mice, which are otherwise called as not-SCID gamma mice, their genetic background is even more intense in a condition where they are not able, where they don't have even the slightest bit of an immune system. There can be conditions where immune cells only lack the B cells, or immune system only lacks the B cells, or the T cells, or the combination of the two, or the combination of the two along with the absence of NK cells, and so on and so forth. Based on the extent to which the immunocompromisation is present, the response of different types of mice models will also be very different. So most commonly we go for the non-SCID mice, like I just mentioned, for implanting tumors or cancer cells, basically to study how they actually grow over a period of time. They cannot be injected into an immunocompetent mouse, which means one which has a functional immune system, because that will help in the removal. It will basically identify that cancerous tumor mass as something foreign, and it will eliminate or clear that from the system. So basically, coming back again to the experiment that was performed, what they actually did was they took a transgenic strain of mice, which here has been marked as A cross B because it is a cross between strain A, strain B, and the F1 generation mouse has been taken for this experiment. That was subject to thymectomy, which means the thymus was removed, and it was subject to lethal X-ray radiation or X-ray treatment or was subject to lethal X-ray, lethal levels of X-ray, because that led to the removal of any T cells that are proceeding towards maturation. So this is a severely immunocompetent mice, which is lacking T cells, functional T cells. Now, when we were able to introduce strain B graft of the thymus, which means that from a mouse which belongs to strain B, they took the thymus and tried to transplant it into this particular mouse with this particular genetic background. And along with that, they also transfected A cross B F1 generation hematopoietic stem cells. So they took another mouse from this same family and took its hematopoietic stem cells and then infected it with an LCM virus with, let's say, any XYZ, any virus with this, just for now. What they now wanted to see was the concept that they were looking at is that if I am say you have a model system from which you're removing the thymus. As a result, you don't expect any T cells. Now you're externally providing T cells to that particular model system. What is the question that they're trying to address? That if I am now introducing these T cells, what do I expect their specificity to actually be visible as? Will these T cells be responsive against both A cross B or only B or only A, or what can be seen? So to address this curious question, the scientists, they took spleen cells and they tried to see LCM infected strain B cells and LCM infected strain A cells. As surprisingly they could note at that point of time, that's in such cases you could not see any killing of the strain A cells. Why? Because it was concluded at that point of time that when you are introducing thymus precursor thymocytes and you're introducing the organ to which it will eventually almost indefinitely almost certainly migrate, which is the thymus, its conditioning will depend upon the surrounding microenvironment and not only on its genetic background. So that means that if you are introducing a strain B thymus into this particular mouse system, its precursor T cells will move towards that side and will reach the thymus. And because the surrounding environment is of a particular manner, they will modify themselves in that particular way. And therefore, they would carry sensitivity, or they would um, they would be able to execute their killing action based on certain strains indicative of what exposure its immune system has already had to. When I say exposure, I mean that whether it has functional immune system or not in this case, just because it's a mouse model. And here, again, this is a representation of the fact that whenever you perform an experiment, you must always have an experimental control. So in the experimental control, they have also taken an A cross BF1 strain of mouse, infected it with an LCM virus, and then they saw that what happened when you are just taking simply an immunocompetent mouse, you are able to see killing of both strain A and strain B cells. However, when it was subject to all of those treatments, what they could eventually identify was that the MHC haplotype of the thymus within which T cells develop actually determines their self-restriction. So basically, when we started off, we talked about the two properties of or the two methods of selection of T cells. So we talked about the positive selection and the negative selection. So the positive selection is actually dependent upon the surrounding environment, because that determines, or let's not say determines, but that very heavily influences the MHC haplotype of the thymus in which these cells are developing. So we'll quickly go over the figure legend. Here you see an experimental demonstration that the thymus selects for maturation only those T cells whose receptors can recognize the antigen presented on target cells with the thymus haplotype. Thymectomized and radially irradiated F1 generation of mice were grafted with the strain B thymus. So basically, you are irradiating these mice and then you are transplanting or grafting them with the thymus of strain B, and you reconstitute it with F1 bone marrow cells. After infection in the LCM virus, the CTL cells are assayed for their ability to kill a particular strain, and depending upon what it has been initially exposed to. So that I'm just not going over this because I think the typing issue has been compromised here, but I want you to understand the takeaway from here. It is the surrounding environment within which the T cell will undergo maturation that determines its MHC restriction, and not the origin of the thymus from which it is coming. So that was the idea here. Sorry. That was the idea here. Now we'll go over this process in a little bit more detail. So again, coming to the flow chart or the pipeline that we had been observing till now, you obviously start off with the T cell precursor, which undergoes rearrangement of the TCR genes and leads to a sort of an intermediate state called as an immature thymocyte. So within the or on the surface of the immature thymocyte, you can see the expression of CD4 and CD8 molecules along with CD3 as well and the T cell receptor. This is this is aligning with what we have already seen in that first schematic representation. So now what happens? You have an immature thymocyte which is double negative in manner. It is undergoing these two processes of selection. You have positive selection of those cells which are only whose receptor is only binding the MHC molecules. What is that leading to? Let's just go over the schematic just to see, be a little more aware of it. What you see here is an epithelial cell. Let's consider it to be a macrophage for now. So this macrophage is presenting antigens via both class 1 and class 2 MHC molecules. In case you don't remember how this antigen presentation pathway works, please go over to the previous discussion sessions, even if you can't listen to the entire video, because I understand it's very long. Please just go over the slides. You will be able to understand, hopefully be able to understand everything, because I've put very simple schematics, and that is what Kubi has as well. You can either read Kubi because it's a fantastic book, or you can at least study, from the diagrams, because that will give you a comprehensive overview of everything you have studied in writing. So basically what you see here is that the antigenic peptides, which are, being presented on the surface of the epithelial cell, which we are considering for now to be a macrophage, you can see that it is being represented by both classes because of the nature of the epithelial cell, which is under question. That is why I said that this can be considered to be a macrophage and not just, say a liver cell or a spleen cell, nothing of that sort. That is because epithelial cells, certain epithelial cells, including macrophages, are able to express both of these proteins and they function as professional antigen presenting cells. So these are the ones that will be allowed to proceed further. So after there's an initial repertoire of, say, just a ballpark figure, that's not even close to the actual figure, but let's say that there are 1 lakh immature, 1 lakh is a bit much. Let's say there are 1,000 immature thymocytes which are present. From there, the first level of filtration or clearance basically is performing positive selection. Any cells that are not able to identify a self-MHC, either one or two or both, that must be removed from the system. Why so? Because if there is no recognition of self, then that means there is no distinction from non-self. And therefore, whenever it elicits an immune response, whatever the reason might be, then that could potentially target the body's own cells. And once it starts seeing a pattern that there is a huge patch whereby this is happening, it can lead to severe disease. So that is what you understand by positive selection. And you can see that all the other non-productive rearrangements have been completely cleared out. So this is positive selection. Coming to the second part, let's move towards the right, which is about negative selection. So negative selection and the death of cells with high affinity receptors for self-MHC or self-MHC plus self-antigen. So what you see here are mature CD4 or CD8 T lymphocytes. And you can see how there has been how an interaction is being studied between this particular thymocyte and the macrophage, or between this thymocyte and a dendritic cell, both of which are antigen presenting cells. The question is, why is the behavior different, or why at all is it necessary? It is because of the way in which you address the problem. Say, for example, you have to make sure that your body or your immune system knows where your body ends and the rest of, where it ends. Let's put it that way. So what you see is that it will be able to distinguish this individual's own system from anything which is foreign. And for that, the positive selection is important so that it becomes restricted only for self-MHC carrying cells. Second, what we see is a slightly more complicated technique, not technique, but a concept. The point is that if you need to ensure that this is your self-MHC, why are you subjecting yourselves to negative selection? it is extremely crucial because the job of a TCR or a T cell for that matter is not to function as an antibody. It's not supposed to go bind to the cell, or even if I'm being a little more precise, then the function of an antibody in a molecular biology experiment, an antibody goes and binds. That is all it is doing. But that is not what is happening here. Here it needs to be in a dynamic state. There should be a turnover of that particular protein and there should be a maintenance which is recorded. So what it effectively does is that it is eliminating any interaction which is so strong that such an interaction is not disrupted at any point of time. Say, for example, if you get an, you know, think of it, say, for example, you meet a friend and while you appreciate them, you do not want to hang out with them every day. This is kind of like that. You want to meet a friend because you know it. And, in this case, the self-MNC is that friend that the T cell knows. And therefore, it should recognize that I know you and you are a part of the individual's body system. And so I will not harm you or attack you or, be defensive in your presence. On the other hand, my interaction should not be so strong that I don't have any other friends or that I end up wasting a lot of my time with just that friend. So any unnecessary, not unnecessarily, but more than usual interaction or more than usual affinity between two proteins can be a dangerous thing. So if that is the case, then such readings also need to be, such thymocytes also need to be cleared from circulation. And effectively what you get is a repertoire of cells that are A, self-MHC restricted and self-tolerant in nature. So coming to the next part. Okay, we won't take very long today. I have not gotten into unnecessary details because not unnecessary, but a lot of details because a lot of it was very memory-based work about the exact cytokines, what they're doing, what is their job, etc. Trying to include more of the associated pathways that you might not be coming across in the course, but that will help you sort of complete the story. So what you see here is T cell mediated signaling pathway or TCR mediated signaling pathway has been, briefly summed up here. So on the figure on the left, you can see how an MHC peptide, this is actually a zoomed in version basically. So you see an interaction between two types of cells. In blue, what you have is a T cell. On its surface, you can see the T cell receptor, that dimeric structure basically. And on the other hand, you are seeing on the other side some other cell, epithelial cell, for example, which expresses class 2 MHC molecule that is carrying your peptide of interest onto the cell surface. Now, first, the TCR and the class 2 MHC molecule must recognize each other. How will it do so? Think of it as these 2 points, right? They are interacting with each other, but the exact entities that are in contact with each other, they, or exact entities that help in the, you know, okay, let's not put it that way. Let's just say there are some points of contact between class 2 MNC molecules and TCR. I don't want to unnecessarily deviate from the idea. So yeah, that's what you see, that when an MNC peptide complex has been, is engaging each other, that initiates the downstream signaling pathway. Once that is done, you also see that there is a co-stimulatory or co-receptor interaction which is occurring. In blue, what you see is CD4 or CD8. They've just been represented here in an interchangeable manner. It doesn't always need to be the case. So what you see is that it is a membrane anchored sort of protein and it is able to interact with the body part of class 2 MHC molecule. And that is a co-stimulatory ligand or co-stimulatory interaction for the same. So yeah, what happens there, we'll just go through the legend for that purpose. So an early step is the LCK mediated phosphorylation of ITAMs on the zeta chain of TCR complex. So you can see this is the these are the different types or different representative types of T cells, and you can see that there is zeta, gamma, delta, and epsilon. So what LCK does, it functions as a kinase. It binds to class 2 MHC and is simultaneously able to trigger or is able to facilitate phosphorylation of proteins. Proteins that are present within the ITAM molecule, basically. Apart from that, you can also see gamma delta and epsilon subtypes, which are present in a different location. Let's put it that way. So a very early step in is the LCK mediated phosphorylation of ITAMs on the zeta chains of the TCR complex, creating sites to which protein kinase ZAP70 attaches and becomes activated by phosphorylation. So what you see on the right is a representation of that part. So you have ZAP70, which is binding to the lipids, and then wait a second, I just messed it up. You are able to see a representation whereby you have this TCRs localized on the cell surface. You're able to see ZAP70. What does ZAP70 do? It acts as a protein kinase. When we say kinase, what it effectively does is that it adds a phosphate molecule and causes phosphorylation of its target protein. In this case, it is able to do so upon this motif, basically, and that was quite helpful. Next, what happens is that CD4 or CD8 associated LCK phosphorylates ITAMs of coreceptors and creates a docking site for ZAP70. ZAP70 then phosphorylates adapter molecules that recruit components of several signaling pathways. Next, you can see what are the different types of pathways. That the details here have not been mentioned, but basically to sum it up, we can see that after there is the initial interaction between class 2 MHC peptide and the T cell receptor, which is being studied, you can see that there is an LCK mediated phosphorylation of the ITAMs present on the zeta chain. Second, that allows the docking of protein kinase ZAP70, what is present here, and its activation is also by phosphorylation. This ZAP70 catalyzed protein phosphorylation is then generating downstream signaling or is affecting or altering the downstream signaling cascades, including PKC or protein kinase C mediated pathways, calcium mediated pathways, small g protein mediated RAS, RAF, RAC mediated pathways. All of these are entirely downstream of this initial interaction. Now, somebody might be wondering, why is that at all the case? Um, it is extremely relevant because after this initial recognition has been facilitated, there has to be an increase in the expression of genes that help in or that aid proliferation and survival, so that helps them to persist over a long period of time. But if we talk about it in a sort of pathway dependent manner, you can see that certain pathways are going up. What are those examples? Starting off, you can obviously see that there is a PKC, like I already mentioned, protein kinase C mediated pathway. You can call any of these people at any point of time. So, just a second. Uh, what you see next is a calcium mediated pathway. Now, what is the reason calcium is being studied here? If you have some background in cell signaling and all of the pathways that are uh, okay, let me use the terminology. There's something called as a secondary messenger inside a cell. What happens is that initially there are some receivers of some signal or some stimulus, which are usually present at the surface of the cell. That is obviously a receptor, cell surface receptor. In this case, that would be the interaction that is occurring between these two proteins. Then there is a signal transducer. Now, many a times, certain proteins or certain receptor proteins are able to transduce the signal themselves by their cytoplasmic tails, or in some cases, there might be some attached protein that is able to transduce or carry forward that signal throughout the cell. Next, you can see that after this has happened, what needs to be looked at is the executor of the effect. So after the initial interaction has occurred, there has to be something which causes a change in the overall cellular machinery. Those are known as secondary messengers. The primary messenger was that signal transducer. The secondary messenger is something whose level goes up or goes down and causes a shift in the overall cellular metabolism. One such very common example is calcium. You have IP3 also as well, that is indicated. Both of these act as secondary messengers, and a shift in their expression or a shift in their physiological levels is recognized as a signal and is of extreme importance. I'll give you one example. Um, Calcium is also Yeah, okay. For example, if you talk about, I think you might have studied it, that there is something called as the sarcoplasmic reticulum, which is basically a part of the in part of the muscle cells, and within it, calcium plays a very, very important role. There is a basal level of calcium which is mentioned, which is maintained, and the movement or the shift in this calcium level from what is maintained at baseline to something which is much lesser, because calcium moves out of those cells, can trigger the movement of a muscle or can trigger muscle contraction, muscle relaxation, and set off that entire cycle. So calcium, a molecule as small as that, is a very crucial mediator of signaling, and it helps in understanding what will happen downstream of the initial process that initiated that signaling pathway. So with this in mind, we understand that IP3 and DAG are just IP3 and calcium are just two stimulators of certain downstream pathways, including protein kinase C. Additionally, we have small G protein mediated pathways. You might also be familiar with that. Commonly, this is understood as GPCR signaling, G protein coupled receptor signaling. As the name indicates, these are some G proteins, globular proteins, which are present on the cell surface. And what they do is they trigger they are 7-pass transmembrane proteins, and they also use similar calcium messengers. In that case, there is cyclic AMP also, which is used as a secondary messenger along with calcium and DAG, or diacylglycerol, which are then able to execute their downstream sort of effects. So, but that is mediated, or that is very heavily dependent upon the G protein which is present on the surface. What is the net effect of all of that? Net effect is that there is an overall change in the expression of certain genes that can differ very widely depending upon what is the cell type, what is the condition being studied, and so on and so forth. But as a result of that change in gene expression, you expect to see some functional changes. Certain genes might help cells to differentiate. Certain changes might help in activation, proliferation, etc. That can, be differ depending on different conditions. So just a second. Yeah. Another signaling pathway that is triggered downstream, or that is associated with T cell activation, involves the phospholipase C pathway, which is PLC pathway, and is activated by phosphorylation. Here you can see there is an inactive phospholipase C gamma, which is present at the cell surface. And the same protein, scaffolding protein that we saw in the previous diagram, ZAP70, is also playing a role here. So when there is phosphorylation of this protein by ZAP70, what that leads to is activation of that protein, releasing the same two secondary messengers, IP3 and DAG, and they execute their own effects in different manners. How so? Let me read with the legend so that it simultaneously helps you in understanding. So phospholipase C, or PLC, is activated by phosphorylation. Active PLC is hydrolyzing a phospholipid component of the plasma membrane to generate the secondary messengers DAG and IP3. Protein kinase C, or PKC, is activated by DAG and calcium. Among the numerous effects of protein kinase C is the phosphorylation of IKB, inhibitor of kappa B, a cytoplasmic protein that binds the transcription factor kappa B, NF kappa B, and prevents it from entering the nucleus. NF-kappa B is a family of proteins basically, but it is usually understood as a transcription factor. It's an umbrella term, but it is understood as a transcription factor. Uh, yeah. So phosphorylation of inhibitor of kappa B helps in releasing NF-kappa B, which then allows it to translocate into the nucleus and then execute its activation, or basically allows it to carry out its transcription and execute its downstream actions. In addition to that, what you also see is that there can be calcium dependent activation of calcineurin. What is calcineurin? It is a calcium calmodulin dependent phosphatase. Phosphatase is something which cleaves a phosphate molecule, or phosphate, which cleaves phosphorylation and removes the phosphate entity. IP3 mediates the release of calcium from the endoplasmic reticulum. Calcium binds the protein calmodulin, which then associates with and activates the calcium calmodulin dependent phosphatase known as calcineurin. Active calcineurin removes a phosphate group from NFAT. NFAT is another transcription factor that allows it to translocate into the nucleus. So that is what you see here represented as well, that the intracellular calcium stores are maintained. However, there is a release of calcium to act as a secondary messenger, and the downstream effects are manifested via NFAT, which is a transcription factor, and that affects the transcriptional activations of genes, which are associated with the which help in T cell activation and its further processing. So initial processes are different. Like the initiator of these effects is different. It is majorly because of an interaction or the recognition of the MHC molecule and the TCR. Once that has occurred, it indicates that this is a particular T cell that needs to be expanded in number, and therefore pro-survival and pro-proliferation pathways will go up, and that will help in in sustenance of that particular variety of T cells. Next, we have an quick, just I think this is the second last slide. We have a quick overview of the interactions that facilitate this activation of T cells. So as you already know that I have not got into details about this because it was already present in the lectures, but basically there are two key interactions that are very relevant to T cell mediated signaling. The first is an interaction between B7 and CD28. What are these? These are surface molecules that are surface antigens or surface molecules which are present on APCs or on T cells. In case of APC, you can see that there is a B7 molecule which is present on the surface of antigen presenting cells. They are able to interact with CD28. Again, CD belongs to the CD stands for the cluster of differentiation, like I just mentioned, and it is a different surface antigenic molecule that is found to be on the surface of CD4 positive T helper cells. These two interact with each other, and what you can see is that CD28 is expressed by both resting and by activated T cells as well. But to prevent unnecessary and indefinite activation of T cells, there has to be a controlling or a regulatory or a maintenance signal that will keep the levels in check. This is facilitated by CTLA4. So while B7 molecules or B7 surface antigen is able to interact with CD28, they are also able to interact with CTLA4. However, CTLA4 binding is not a positive interaction and instead keeps a check on how much T cell activation is allowed to proceed. Therefore, in some cases, like if you see a disease condition, say for example, where CTLA4 expression is going up on the surface of the helper cell, then that will outnumber the number of CD28 molecules that are present, and that will eventually lead to a condition where T cell activation and T cell mediated signaling will be brought down to a minimum. So that is how these two interactions work, and they are antagonistic in nature. What is the effect of it? One of the common effects has been demonstrated on the right. You can see that the interaction between the B7 and CD28 molecules present on the APC, normal APC, and the particular cell of interest which is being studied, which is the T helper cell. You can see that when there is an interaction between these two, signal 1 is coming from the TCR and the MHC molecule interaction that is allowing the trigger, that is giving the first how should I mention it? The first message that this is a functional or this is a productive interaction between two types of cells. When that has been recognized, a second signal, a supportive, additive, or a secondary signal is occurring between the B7 and CD28 molecules, which is allowing the synthesis of the IL-2R gene. Both IL-2 and IL-2R genes are then expressed that allows them to interact with each other and then execute its downstream effect, which means that there is a release of IL-2, and IL-2 is basically an interleukin which functions as a cytokine and then is allowing it to execute its action. The question is, why is that relevant in this case? This is relevant because while IL-2 that is normally secreted by these cells, because of the basal levels of B7-CD28 expression, is able to execute its activity in a paracrine manner, which means that it is able to execute its activity on other nearby cells. However, when there is a co-stimulatory signal that helps the pre-existing signal, that is by interaction between B7 and CD28, that helps in the expression of IL-2R gene, which is a receptor present on the cell surface. When that happens, what is it doing? It is allowing an autocrine fashion of signaling to be now put into place. This means that the cell is able to produce its own ligand and produce its own receptor, and therefore sort of executing a self-sufficient job. It is causing autocrine signaling to drive its own activation and own proliferation, so that it can expand in numbers and do so in a very large scale manner, generating an entire population, like you see just beneath that in the schematic as well, that you can see an entire population of cells which can then execute their activity either as memory cells or as effector cells, so that they can sustain within that individual circulation for a definite period of time. So that is the entire job. And apart from that, within this diagram also, you can see that the IL-2, IL-2R receptor interaction is driving certain cell cycle genes as well, which is pushing them to proceed more and more towards proliferation and expansion, clonal expansion. Basically, that means increase in their numbers, and they are able to generate that entire repertoire for that purpose. Coming to the last topic, this you might already be familiar with, but I wanted to revise it very quickly before we move to the questions that were given as assignment for this week. We will be discussing the assignment directly because its deadline is already passed, and I'll also go over some PYQ so that it is better for your understanding. So yeah, so this is basically a comparison between all three and their response to with respect to MHC expression, their ability to activate T cells, and basically a comparison of their overall functions. So if you remember, we had the three antigen presenting cells, dendritic cells, macrophages, and B lymphocytes. You can see what types of proteins are expressed on their surface. So for dendritic cells, we have B7, we have class 2 and class 1 MHC. For macrophages, you are able to see two different states. Either it is a resting macrophage or an activated macrophage. If it is resting in nature, it only expresses class 1 MHC on its surface. However, once it has been activated, class 1 MHC is co-expressed with the help of course, LPS and interferon gamma, which are stimulatory molecules. We will learn more about them and how they are relevant towards their activation eventually, along with cytokines, I think. And what it effectively does is that along with MHC class 1, you can see MHC class 2, and also B7 expression on the surface of activated macrophages. Third, you have a population of B lymphocytes, which are either resting or activated in nature. They have expressed class 1, class 2 MHCs because they're antigen presenting cells. They are also professional antigen presenting cells. They are also co-expressing class 1 MHC. The difference between them being the fact that when it is an activated B cell, then it also co-expresses B7 on its cell surface in comparison to the resting state. Now, for each sub-condition, whether it is resting or activated, a comparison of their attributes and their responses have been made up. When we talk about antigenic uptake for dendritic cells, we have endocytosis and phagocytosis. Specifically, phagocytosis is for the Langerhans cells, which are also a variant of the immune cells of the dendritic cells itself. Their class 2 MHC expression is constitutive and is quite high, which is what you can see by the presence of 3 plus signs. That means they have high level of dendritic cells have a high level of expression of class 2 MHC molecules. Third, you see that there is a co-stimulatory activity by B27, which is present. B7 and CD28 are executing their activity or their co-stimulatory activity. And T cell activation can be carried out by naive T cells, effector T cells, or memory T cells as well. For macrophages, we are seeing that resting state macrophages can take up antigens by phagocytosis, which is then supported by their activation further they are not inducible, they don't have inducible B7, or they do not have T cell activation capacities, or they have very basic levels of inducible B7 activities. But activated cells, activated macrophages are very, very much capable of taking up their antigens by the process of phagocytosis, but instead they also have inducible class 2 MNC expression. What you need to keep in mind is that there is a difference between the term inducible and the term constitutive. Constitutive means that dendritic cells will always keep on expressing class 2 MHC molecules. On the other hand, macrophages lack that or not some cytokines are present. Similar response is also seen for B7, whereby its expression is also found to be inducible for macrophages in this case. And along with that, T cell activation is possible, and it only works on effector T cells and memory T cells, but not on naive T cells, which means that T cells that have not yet seen their antigen, a cognate antigen, cannot be activated by macrophages, whereas dendritic cells are able to do so. Third, T lymphocytes in their resting and activated state are being compared. So you can see that both these subtypes of B cells are able to take up their antigen by receptor-mediated endocytosis, which means the receptor is allowing the process of endocytosis to be executed. Class 2 MNC expression in both cases is still constitutive, which means that it is doing a better job of expressing class 2 MNCs than macrophages. However, its B7 expression or B7 activity and expression both are inducible in nature. This is where it becomes less potent than the not potent, but let's say it's less efficient than the job that dendritic cells can do. Finally, you see T cell activation can be performed for resting state B cells. It can only be performed for effector T cells and memory T cells. Whereas for activated B cells, they're able to do so for all three types. So while activated B cells are almost as good as dendritic cells as antigen presenting, with respect to their antigen presenting capability, they still do not have a constitutive expression of B7, thereby making dendritic cells the most potent, most efficient, and most abundant, not abundant, but yeah, most potent antigen presenting cells, or the most most potent, let's put it that way. So this is how the comparison basically ends. And with this, we come to the end of the presentation. I will quickly go over the assignment questions, and then we can, sorry, just a second. Then we can wrap up the session for today. If there's a doubt, please let me know. I know some of the topics were a little vaguely discovered, but I didn't want to get into unnecessary detail because most of it has been covered and I knew it would be difficult to accomplish within this brief time. But yeah, please let me know if there's some topic you want me to discuss on Sunday, so I'll try to include slides based on. Okay, so yeah, so this was the assignment. These were the assignment questions for this week. So the first one was about the nude mice, which we have discussed. What do they not possess? They do not possess bone marrow, do not possess thymus, B cells, or none of the above. The correct answer in this case, of course, is option B. They are thymectomized mice, which means that they do not possess a functional thymus. Second, what are double negative T cells? T cells that are both CD4, CD8, do not have CD4 but have CD8, do not have CD8 but have CD4, have neither. So obviously, as the name suggests, this was again a very, very direct question. I hope everybody was able to get it right. That means these are those subtypes of cells that do not express either of the two surface antigens on their surface, which means they're CD4 negative and CD8 negative as well. Coming to the third question, the T cells and B cells share common A, place of development, B, origin, C, set of surface receptors, or all of the above. Now, while I have not explained it directly, if you have listened to the lectures, you would know the answer to this. The origin of both types is coming from the thymus. Their maturation occurs at different sites. Maturation, actually the site of maturation determines how they have been named as well. So while B cell maturation is occurring in the bone marrow, T cell maturation is occurring within the thymus, thus giving rise to the difference in their names and the difference in their let's say, specificity to some extent. Not specificity, but their mode of action as well. So the correct answer in this case was origin, that is option B. Then coming to question 4. During development, if the T cell receptor interacts with MHC1, then it would become a dash. So you are saying that if it so happens that TCR is interacting with MHC1 versus if it is reacting, not reacting, but interacting with MHC2, what do you expect to be the difference in the downstream uh, effect of it? So basically, if a TCR interacts with MHC1, it would become a CD8 positive T cell. It would become a CD8 positive T cell, and if it does so with MHC2, then it would become a CD4 plus T cell. So that is again a direct answer, option A. Then we were asked about the correct sequence of T cell activation in lymph nodes. Um, sequence of T cell activation within the lymph nodes. What was the correct order of arrangement in this case? So we'll read the options quickly. First is that activated T cells differentiate into effector T cells and they exit from the lymph node. T cells enter the lymph node from blood via HEV. Uh, whether they T cells not activated by antigens exit the lymph node. Activated T cells. Activated T cells lose the ability to exit from the lymph node. So of these, the correct option is highlighted as option C. So the sequence of events that if we follow the same track would be from 2, 3, 4, and then 1. So it starts off with the fact that T cells are entering the lymph node from blood via the high endothelial venules. Second is that it is T cells not activated by antigens. They are exiting the lymph node and then eventually would be cleared from circulation as well. Fourth is that activated T cells will lose the ability to perform this exit. Only the ones that do not interact will be able to exit it and will be able to remain in circulation until they're eventually cleared from the system. But once activated, they are no longer able to exit that site and should stay there and should wait for interaction with more and more concrete antigens. And then comes activated T cells. They will be able to further, after interaction with the cognate antigen, will be able to further differentiate and form memory T cells and effector T cells because of the multiple interactions that I just talked about between T7, CD28, and also the MHC and TCR interactions, which allows the expression of many genes, including the IL-2 gene and IL-2R gene, thereby activating autocrine signaling and sustaining cell proliferation and its differentiation into memory and effector T cells. And coming to question 6, which of the following interactions are required for clonal expansion and differentiation of naive T cells? So option D would be correct because all of the options are, I'll go over it even though quickly. So for naive T cells to eventually undergo expansion and differentiation, the first interaction that obviously we are most familiar with is the one between P7 and is the one between MHC and TCR, which is option A. Second comes the co-stimulatory interaction that I just mentioned between P7 and CD28, which is helping in validating the accuracy of that interaction even further. Third is the binding of some cytokines to the cytokine receptors. In this case, the example we have studied is IL-2 binding to IL-2R. So that is the third stimulatory signal. And together, all of these will be making option A the correct option in this case. And if you would just to keep it in your mind, that CTLA4 B7 interaction in this case would not be a stimulatory interaction, but would instead quench it and prevent further differentiation and proliferation of those T cells. Next, we were asked about T cells in the secondary lymphoid organ. They are assembled by CCL21, CCL19, CXCL13, and CCL13. So while you don't necessarily need to remember all examples of all different, surface receptors or even cytokines for that matter, This was a very fact-based sided question. There's no way to bypass it. Some examples, if you have on mind, it will always give you an edge over others who might not necessarily have paid so much attention to it. So it was just a very direct question, and that was the correct answer here. That was CCL 19. There are not any tricks or tips for you to get by with this. It's a simple memory based question. If you remember it, you'll be able to answer something like this. Then we've been asked that if T cells enter the lymph node cortex via which of the following: cortical sinus, efferent cortical vein, or high endothelial vein. So we have already discussed it in a previous question, just 2 3 questions back. And HEB was the correct answer in that case, that through high endothelial venue, they'll be able to enter the lymph node cortex. Ninth is about diapedesis. So diapedesis is a movement of lymphocytes through the endothelial wall, movement of T cells from thymic cortex to thymic medulla, movement of B cells from dark to light zone, or rolling of neutrophils on the surface of the endothelium. So this is simply the movement. Diapedesis is the movement of lymphocytes through the endothelial wall. If you remember the schematic that was shown by Professor Shudip in the lecture, it actually demonstrated how they're able to and sort of squeeze themselves through the uh, gaps between the cells of high epithelial venules and move towards the other side. So yeah, that is how that is the correct answer. And finally, LFA1 on T cell surface binds to antigen presenting cells through which of the following: CD28, CD58, CD4, and ICAM. So I have not shown it in my slides, but yes, if you go through the last lecture of Professor Shrudeep, you'll be able to answer this question because there are multiple subtopics that he has covered regarding the same. So LFA1 on T cells is able to actually interact with ICAM molecules present on the surface of APCs or antigen presenting cells. And therefore, option B was the correct answer in this case. So with this, yeah, I think I've covered everything I wanted to cover for today. So if there is a doubt, please go ahead and mention it. And if not, then we will conclude the session for today. Okay, if not, then I'll conclude the session for today. Thank you for joining as per our regular schedule. And I'm very sorry about the last 2 weeks. There has been some disruption, but it has been sorted for now. And yeah, we should be able to get back on track and have more interactive sessions and try to address queries in a much better manner. So thank you for joining and have a wonderful night. Good night.